The title of my talk today will be Comparative RNA Sequencing Analysis for Children with Cancer, and I will explain what comparative RNA sequencing means. We do focus, so as Ashley mentioned in the introduction, we have this Treehouse Childhood Cancer Initiative that we started at UC Santa Cruz, so we have a big focus on pediatric cancers. However, the approaches that I will be discussing are applicable to other cancers as well, and also potentially applicable to rare genetic diseases. At the end, I can make some comments about how these approaches could be used to help identify the molecular cause of undiagnosed genetic conditions. So RNA sequencing, just very brief background, is a high throughput sequencing technology. So typically the most common sequencing platform that's used today is the Illumina sequencing. This technology measures transcript sequence, but also the abundance. I'm sorry about that noise. It's gonna stop, I promise. It's some calendar thing. So RNA sequencing can detect multiple RNA populations, including messenger RNA, non-coding RNA such as lincRNA, microRNA, or tRNA. For clinical applications, I think mRNA or protein coding RNA is what's been most studied and what would be mostly used in the clinic. But these other types of RNA are also important, and maybe later, as we learn more about these various species, they will become important in the clinic as well. So in cancers, RNA sequencing can be used to detect gene fusions, which are chimeric products that are produced by DNA translocations. You all have heard of BCR-ABL fusion. So things like that can be detected by RNA sequencing. RNA sequencing can also be used to detect express sequence variants, so both SNVs and indels. And it's important sometimes to know which variants are expressed when you're trying to distinguish between driver mutations in a tumor and a passenger mutation. RNA sequencing can also detect splice variants. And finally, it can be used to measure gene and transcript expression levels. And so this last application is what we have focused on. So I feel like in the clinic, there are already assays for gene fusions and for mutations that others have addressed, but this really, the application of RNA sequencing to look at gene and transcript abundance is not as widely addressed, especially in the clinical setting, and that's where our work has focused on. So RNA sequencing for gene expression applications. So RNA sequencing, sometimes people call it a tag-based approach, where basically what you're doing is you're counting the number of reads that map to a particular exon. So I have a gene model here with various exons in different colors, and then introns in gray, and then these in black are sequencing reads that you can align back to the reference, and then you can quantify how many reads align, and so that's how you can derive a quantitative estimate of expression. Usually the measurement that's used commonly in the field is transcripts per million. So then these read counts can be normalized to the total number of reads in a sample, and also to the length, to get something like transcript per million measure that can be used to compare genes to each other. So the expression of one gene to the expression of another gene. So RNA sequencing, especially so this application that I was talking about for expression levels, it's expression levels are only meaningful in relation to something. So it's a relative estimate. Something like a gene could be highly expressed or it could be lowly expressed, but it's only meaningful in relation to a reference. And so we believe that that's why, that's one of the limitations for why this application has not been as widely used as some of these other applications of RNA sequencing. And since this is a relative measurement, having access to data from other specimens to compare these expression levels too is crucial. And so the problem that there is in the field is that the genomic data are siloed and not shared. So individual laboratories and individual hospitals are generating data, and kind of that data remains usually in that laboratory or that hospital because there are a lot of issues. There are both technical and regulatory issues with sharing data. However, if the data are shared together, then there is more data sets that could be available for comparison, and these relative gene expression measurements can be more meaningful. And so with that in mind, I will share one clinical anecdote for how our Treehouse initiative was started, and why I talked about data sharing, and why I think data sharing in the RNA sequencing space in particular is very important. And it's important to note, I come from UC Santa Cruz, and UC Santa Cruz does not have a medical school or a medical center. So for us, this experience that I'm going to be describing was very unique. And so it started with an 8-year-old boy who was diagnosed with an undifferentiated CNS sarcoma, and this patient was actually in Canada, so it was a Canadian patient. As Ashley mentioned, I did my PhD with Dr. Mark O'Mara, and so this patient was actually treated in Vancouver, British Columbia. And so was a patient with undifferentiated sarcoma, then treated very aggressively as these sarcomas would be treated. And then 2 years later, the patient relapsed with metastases to his lungs. So then there was no standard treatment options available at that point, and the patient enrolled on a clinical genomics trial, which characterized his tumor DNA and RNA through sequencing and revealed this gene fusion, EWSR1, ATF1 gene fusion that's common in sarcomas. However, as a result of this, there was no therapies identified for this patient. And so, back to square one, what do we treat this patient with? And so on a research basis, the data from this patient, the RNA sequencing data in particular, was shared with our group at UC Santa Cruz. And we did this analysis where we compared, and I'm going to describe the analysis in more detail later. But what we essentially did is we compared this RNA sequencing profile from this patient to RNA sequencing data from other cancers that we assembled. And the reason that we had data from other cancers that we assembled at UC Santa Cruz is because UC Santa Cruz was heavily involved in the Cancer Genome Atlas research initiative, and also was at the time the repository for all the cancer sequencing data from this initiative, the Cancer Genomics Hub. So we had a lot of cancer data at UC Santa Cruz, and kind of on the research basis, we did some comparative analysis. What we also identified in this sarcoma is so I'm showing a molecular pathway. This is the EWSR1, ATF1 gene fusion at the top. And then some of these genes are colored in red. So the genes in red are genes where we saw that their expression from the RNA-Seq analysis was in the outlier range, so was very high in the sarcoma tumor. And I will explain the outlier analysis later, but suffices to say that we saw overexpression of several of these various genes in this pathway. There was also DNA analysis done on this tumor as part of the study. And so we were also able to map, with our colleagues in Vancouver, were able to map copy number information. So for example, this patient also had an amplification of NTRK1, and also NTRK1 was overexpressed. So there was some DNA data that could be mapped on this pathway as well. But the interesting thing that emerged is in this pathway, there was a particular molecule, JAK1 kinase, that was expressed really highly in this patient. And so if we look at the normalized expression, gene expression on the y-axis, and then on the x-axis, various tumor categories. So these are all of the lung cancers, and we compared to lung cancers because this was a lung metastasis. And then these are all sarcomas because this was a sarcoma, so we compared to all sarcomas. This is all cancers, and this is patient one. So in patient one, JAK1 expression was really through the roof. So it was seemed to be unique to this tumor. And so then from there, because there was no other treatment options available for this patient, the oncologist in the case decided to treat the patient with ruxolitinib, which is a single agent JAK inhibitor, and JAK inhibitor is approved for some leukemias, but had not been used in sarcomas in this setting as in this patient before. And so this is this graph shows the response of the patient, and we have response in terms of landscape score and also the weight of the patient. And I'm not a clinician, so probably people in the audience know much more about all these measures, but from the clinical side, there was a response to the single agent ruxolitinib, which was, and in fact, so there was tumor started responding, and then ruxolitinib stopped, and so you can see that the patient was at the beginning at score 60, but then very quickly his performance improved, and then ruxolitinib was stopped, and then the patient deteriorated a little bit, and then his mom actually wanted the doctor to restart ruxolitinib because she felt subjectively that the patient was doing better on treatment. And then ruxolitinib was restarted, and then there was another response before the patient unfortunately passed away to disease. But what was remarkable was that there was some response to this single agent therapy in the context of a relapsed tumor that was aggressively growing. And so the clinician in that case thought that this analysis was actually helpful for the patient. For us at UC Santa Cruz, this was remarkable. As I mentioned to you, we're not a medical center. We're not used to kind of patient cases like this. And so we got inspired by this, and we wanted to ask the question, well, can we assess this in more patients and see if this type of analysis could be useful for other patients, or is this really a one-off anecdotal example? And so with that in mind, we started this Treehouse Childhood Cancer Initiative. So the patient's name was Kelvin, and he wanted his name known. He actually asked for his name to be used in presentations rather than patient one, and so I'm honoring that wish. And so Treehouse was started because of Kelvin, and the goal of Treehouse was to leverage big data sets, so these large shared data sets, for identifying new targets for difficult to treat pediatric cancer cases. So Treehouse relies on cross-tumor RNA sequencing comparisons. So as I mentioned, we compare individual RNA sequencing data set from a tumor to data from many different tumors. And we have a hypothesis that such a comparative analysis can be useful for identifying unusually expressed oncogenes, as we saw in this case, JAK1 was unusually overexpressed. And we also further hypothesized that this analysis would improve the more data we have, because then we would be able to compare it to more cases and really highlight unique overexpression events. So with that in mind, we started Treehouse Childhood Cancer Initiative. This project started back in September 2015. Initially, we started as a consortium involving UC Santa Cruz and then several pediatric oncology sites that were conducting this type of clinical trial, doing genomic analysis of tumors and trying to use that information to guide treatment. So we've partnered with Pacific Pediatric Neuro-Oncology Consortium, focusing on brain tumors, Children's Hospital of Orange County, and Stanford University. And so all of these partners were doing some sort of clinical genomic trial, as part of which they were generating RNA sequencing data on these difficult to treat cancers. And so the goal of this was to research the feasibility of doing comparative RNA sequencing analysis that I described for our first patient, but systematically with these multiple partners and in a prospective setting. So for patients that are being treated on these studies. And then later, and so this was started with funding from the state of California. This was our governor's initiative to advance, California Initiative to Advance Precision Medicine. And then later we were lucky to get funding from St. Baldrick's Foundation to increase this program and to add some more sites, including UCSF and Nationwide Children's Hospital, and then also the POETIC Consortium of Clinical Trials, and the Profile Initiative, which is a Canada-wide initiative. And so again, all of these partners are conducting clinical genomic studies in pediatric cancers, and UC Santa Cruz, so our role is to get RNA sequencing data from all of these studies and perform the analysis that I described, and then communicate the results back to these partners. So I talk a lot about comparative RNA sequencing analysis and comparing individual cases to compendia of other cases. This is a visualization of what we call our tumor RNA sequencing data compendium. So here, every small dot represents an individual tumor RNA sequencing data set, and then the dots are laid out according to similarity of their RNA sequencing profile, and then color-coded by diagnosis. And this kind of provides a visual representation of the data we have. And the data comes from various different studies, including large studies like TCGA and Target, but also small individual studies. We currently have over 12,000 tumors, over 2,000 pediatrics. So we also have adult tumors in here, as you can see from the diagnoses. We have a couple of other compendia that we've built. So this is a compendia of RNA sequencing data generated using the polyadenylation protocol, poly-A selection protocol. But then we also have a ribodepletion-based compendium. We also have a cancer cell line compendium and a normal tissue compendium that could be used for these types of comparative analyses. And then we use these compendia. So one way we use these compendia is to look for these overexpressed cancer driver genes or overexpressed oncogenes like JAK1. And we use this gene expression outlier approach, where if we look at the gene, so here we're looking at the gene called ALK, which is important in some pediatric cancers like neuroblastoma. And so here on the y-axis is the number of samples in our compendium, and on the x-axis is the expression level of ALK. And here is outlier range. So we use statistical definition of outlier to define this yellow range, where if we have a sample where ALK is expressed in this yellow range, then we call it a gene expression outlier, and we think that that might be significant. And then we can perform this outlier analysis comparing the gene expression in a given case to all tumors. So we call it pan-cancer analysis here at the top. Or we can compare to similar tumors. So for example, if we have a glioma patient we're analyzing, we could compare that glioma to other gliomas in our compendium. And these two analyses could be useful for different reasons. We usually do both, and we get excited if there is overlap. For example, if something is an outlier in both approaches. And then what we can then also do is we can look at these outlier genes we have defined, and then we can look for pathway features. You know, are there multiple genes in the same biological pathway that are affected together, or overexpressed in concert or underexpressed in a sample together? And so that could be useful for strengthening our reasoning that the tumor is dependent on that particular gene. So because we aim to use this analysis to communicate the results to clinicians and for them to potentially use this for clinical decision making, we really spend a lot of time thinking about the quality of our analysis. And this is especially relevant because, as I mentioned to you, our compendium has data from multiple different sources, including research studies, clinical trials. And so data are coming from multiple different sources. So how do we ensure that the quality is okay for us to derive some meaningful insight? And so we developed a metric that we call mapped exonic non-duplicate reads. So really looking at how many reads in each RNA sequencing data set map to exons and are not duplicated. And we've shown that that measure is a good measure to have for each data set. And then we require So don't worry about the technical details of that. If somebody is curious about how that works, I can talk about that later. But suffices to say that for each data set, we calculate how many manned reads are there, and then we have some cutoffs that we use in order for us to trust the outlier analysis, for example, from that data set, and also whether to include that data set into our compendium or not. So we have this quality control. So here is a diagram of our whole process. As I mentioned, we are not a medical center ourselves, but we work with other clinical partners that conduct these genomic characterization trials in pediatrics and generate RNA sequencing data. The data is generated at the clinical partner sites, and then we get the data at UC Santa Cruz, and then we do the analysis. So in gray is the things that are happening at UC Santa Cruz, and in blue are activities that are happening at clinical partner sites. And so at UC Santa Cruz, we do our comparative RNA sequencing analysis. We look for outliers. I'll talk about most correlated samples, so that's another analysis we do. And then we look for overexpressed druggable pathways. And then we meet together with our clinical partners in tumor board meetings where we present our analysis, and then also hear the clinical perspective about these patients, and then have discussions about how these results could be potentially useful to the clinician in their decision-making process as to what to do with these difficult cases. And this is a summary kind of from our initial study that we just published last year, summarizing our first cohort that we did this on of 144 samples. So we generate a research report that we produce and provide to clinicians. So it's important to mention that our approach is not a CLIA-approved test. So it's currently a research assay, although we have ambitions for this to become a clinical assay. And so we are developing and putting in place kind of the QC framework that a CLIA assay would need. You know, one problem is we don't have a CLIA license at UC Santa Cruz, so we have to apply for it, which we intend on doing. So for right now, we generate a research report that's for research purposes only, and this is aimed at clinicians. So this is for the clinician to, you know, perhaps get some useful information about this tumor. So the report includes kind of clinical information, also any molecular testing that was done, so foundation medicine, any mutation testing. We do take that into consideration when we interpret our information as well. And then the other two things, so two important pieces of results that we provide. Number one is what are the tumors that are most correlated to this case, and this could be useful in diagnosis. So I will talk about a case later where this information was useful to refine a diagnosis of a rare tumor. So we report this. So what are the most similar tumors to our case? And then we report visualization on our tumor map. So I showed you the tumor map before that shows kind of our overall compendium and all the different cancers. And then we can highlight on this map what are the most similar cancers to the cancer in question, to our focus sample, and then we can highlight on the map. So in this case, they're all Ewing sarcomas. There is one Ewing sarcoma here. So all of the Ewing sarcomas are here, most of these similar cancers, and then one of these similar cancers is in another sarcoma cluster, but it's still a Ewing sarcoma. So perhaps not so exciting in this case, but I will show you another case where this is exciting. And then the second thing we report is a candidate cancer driver pathway. And this is where we look at these gene expression outliers, so uniquely overexpressed genes. And then a human analyst reconstructs this pathway manually from, you know, considering the literature and based on their cancer knowledge. And then some of these genes that are outliers could be considered as potential drug targets that we could discuss with the clinical teams. We also, so we've been working very closely with Stanford on this, and one feedback that we got from the Stanford clinicians is they wanted us to provide some sort of a score of how confident we are that this outlier that we are finding is a good biomarker of treatment response to a therapy that would inhibit that outlier gene. And so we developed this system that we call RNA Ranker that's aimed at evaluating both the strength of analytical evidence, so how sure are we that something's overexpressed, but also literature evidence, has overexpression of this gene been reported to be a biomarker of response to therapy that would target that gene or not? So that's literature evidence. And also similarly, has this been reported to be a biomarker of resistance to therapy? And so then we can also present this information to the clinical team. And then we submit all of our interpretation to the CIVIC database, which is a public effort where many different experts submit their interpretations, and this could be reviewed by experts and refined as well. So all of our gene expression interpretations are in CIVIC. And so this is the kind of thing that we would report to the clinical team. So for example, in this case, we found 4 different genes that are overexpressed, and all of these genes could be potentially targeted, or all these proteins could be targeted by a targeted therapy that's available. And then we can use analytical evidence, so that's the strength of overexpression, whether something is an outlier, whether there's pathway support, but also literature evidence in terms of has overexpression of FGFR3, for example, been reported to be a biomarker of pazopanib, and what's the evidence for that? And then we score these literature evidence, and then counter evidence. Has overexpression of FGFR3 been reported to be a marker of resistance to pazopanib, for example? And so that's something we can discuss with the clinical team. So then I will show you 2 cases where this analysis has been useful in the clinical setting. The first case is a case of a 10-year-old girl with immature teratoma with metastases to the pericardium and diaphragm. This was a Stanford patient. She was treated with surgery, and then the pericardial lesion was subjected to DNA sequencing and RNA sequencing. And RNA sequencing was analyzed by us. What we found, so this was a teratoma tumor, and then when we did our molecular similarity analysis, we found, and so you can see in the table that the most correlated samples were actually glioma tumors, so were actually brain cancer. And when, so a graduate student did this analysis, and when she did it, we first were like, oh, there must be some error here, this does not seem reasonable. Then we looked into it, and indeed, this teratoma was very similar to brain tumors, to gliomas. We looked at the expression of specific glioma markers, and indeed, this teratoma tumor seemed to express markers of glioma tissues. So then pathology, so then by histology analysis, and you guys in the audience have a much better interpretation, can interpret these images much better than me, I'm sure. But what I'm told is that indeed there are features of glioma in this tumor. And so then based on this analysis, this tumor, the diagnosis was refined to teratoma with gliomatosis peritonei, which is a rare entity where mature glial tissues in peritoneum can occur together with teratoma. And this is difficult, apparently, to identify via histopathological analysis alone. And I'm sure you guys know much more about this than me. So 2 years post-resection, this patient was healthy. And this is a case report we published where essentially this comparative RNA sequencing analysis was useful for diagnosing a rare tumor. So then I wanted to discuss another case where our analysis was useful to identify treatment targets. So this is also a Stanford patient. So this was a 1-year-old boy diagnosed with myoepithelial carcinoma of the liver. This is also a very rare type of pediatric cancer. This tumor was I deficient and showed SMART-B loss, which are biomarkers that have been reported in pediatric cancers. So this patient was treated with chemotherapy, and then 3 years later, the tumor recurred in the lung. And then again, at that point, this is a rare tumor. There was no standard of care after relapse, and no clinical trials for this disease because this is a very rare entity. So then our analysis, so then we perform RNA sequencing and analysis of this tumor. And so first the tumor map analysis. So this analysis again gets at molecular similarity. What is this tumor similar to in our compendium? And so we found that it was similar to kind of other myoepithelial carcinoma tumors that we had, which was, reassuring, but nothing kind of clinically actionable from this. But then from our outlier analysis, so looking for overexpressed oncogenes and overexpressed pathways in this tumor, this is the pathway we reconstructed in purple. So this gene was an outlier with respect to all cancers and with respect to similar cancers. And then in orange, these are all outliers with respect to similar diseases. And so there were two interesting treatment targets in this case. So pazopanib is an RTK, so receptor tyrosine kinase inhibitor, and it can target multiple receptor tyrosine kinases that were overexpressed in this case. And then ribociclib is a cell cycle inhibitor that can target cell cycle CDK4 and CDK6 cell cycle cyclin-dependent kinases that appear to be overexpressed in this case. So those were two kind of takeaway points from this analysis. So, and actually, so first the patient was treated for 3 months with pazopanib. So our Stanford colleagues tell us that pazopanib is, one of the few agents that in pediatric solid tumors, especially sarcomas and some of these other tumors, that pazopanib is kind of a go-to sometimes for these patients. And our analysis suggested that this might be useful. So for 3 months, the patient was treated. There was no evidence of disease at primary site, but then a growth of 2 existing nodules and 1 new nodule was observed, and pazopanib was discontinued. And then after that, the patient was treated with this prediction, ribociclib. And this has been a remarkable story for us. So far, the patient received 12 cycles of single agent ribociclib for over 1 year. And then repeated CT chest scans shows stable nodules. And then eventually, so after 1 year, the oncologist was comfortable enough to say, okay, let's maybe surgery could be indicated to remove these nodules. And so following surgery, the patient is now no evidence of disease, and continuing on ribociclib maintenance treatment for 6 to 12 months. And notably, there is no side effects for this child, so this therapy is really well tolerated, and the patient right now was able to resume kindergarten, although with COVID, now they have to worry about that. But this was a remarkable case where in a relapsed setting, the patient responded very well to this single agent therapy that targets an overexpressed gene. And so we're hoping, of course, that he'll continue to do well. So in summary, I hope I have shown you that sharing genomic data sets can have a clinical impact, and there is potential for using RNA sequencing approach for gene expression analysis and for gene expression comparisons, and there could be some clinical benefit. And in particular, I have shown to you cross-tumor comparative RNA sequencing analysis that we developed, and 2 applications that I've shown is that it can be used to help refine diagnosis, and this is particularly helpful for rare tumors or tumors with ambiguous histology that are hard to diagnose otherwise. I've also shown you that this analysis can reveal overexpressed genes and pathways, and some of these genes could represent druggable targets that could be useful therapeutically in the patient. So what are we doing next? So we have all these data, and so on the research side, we can mine these data to derive new biological insights. We're also investigating what are the molecular mechanisms of outlier expression, so why some of these genes that we see are overexpressed. Like in the first case that I showed, the JAK1 overexpression, we still don't know why JAK1 was overexpressed. Also, so the latest application we have in the lab is that we're developing germline RNA sequencing analysis. And so there's been already several papers of trying to use RNA sequencing in a setting of undiagnosed genetic disease. And so patients, for example, that have a complex diagnosis and suspicion for a genetic etiology, then they often would go for whole exome sequencing or sometimes even whole genome sequencing. And then that exome is negative, for example. So where do you go from there? And so one approach is to do RNA sequencing of that patient. So either the blood, or if there is affected tissue available, for example, for neuromuscular conditions, if there is a muscle biopsy that could be done, so then RNA sequencing could be done, and then this outlier concept that I described for cancers could be done to identify genes that are uniquely overexpressed or uniquely missing in that patient, and those genes could be useful to consider for a potential etiology, especially if you identify a gene that's known to be involved in a condition with a similar phenotype, then maybe, you could look for genetic reasons for why that gene is over or underexpressed. And this approach could help interpret variants of uncertain significance. So I don't have a lot of data to show you for this application, but I think that this is another useful application for comparative gene expression analysis from RNA sequencing data. And to enable this application, we're actually building compendia of data derived from non-cancer tissue. So the normal tissue compendium has data from normal human blood from many different individuals, so that we can have a compendium to compare to. So hopefully sometime soon I will have some data on this. And then in summary, or in conclusion, I just wanted to thank my members of my lab and members of our Treehouse initiative and our clinical collaborators who are actually brave enough to talk to us and to consider our results and give us very important feedback. And then we have a number of clinical partners I mentioned, and also our funders that fund our important work. Thank you. So I'm a little early, but hopefully there are questions that I can address.
Ashley, you're muted.
Sorry, guys. Thanks, Elena. It was an excellent talk. We have a question from the audience. Dr. Cookson wants to know, what kind of turnaround time from tissue to analyze data do you think is achievable?
Yeah, that's a great question. And that's something so when we started this project, it was a couple of months turnaround time, which is not, we've had a lot of patients where this was too long, and so we've shortened. So on our analysis side, we can do 1 week turnaround, but then there are other aspects of this, the pathology, so obtaining the tissue and then sending it for sequencing. And so we currently work with a lab that does RNA sequencing, Covance or LabCorp. So they're a CLIA lab. And so the logistics of, you need to send the sample there. And then sometimes they're fast and sometimes they're not. And sometimes it's not in our control. On the analysis side, once we have the data, we can do 1 week or even less. I would say right now, the whole process, I'd say, is 1 month turnaround, but we know that that's too long. We're definitely working to shorten it. Thank you for that question. That's a really key question to all of this, is how quickly you can do it. Then the other thing we're thinking, so we've been doing this analysis for difficult to treat patients that have been through, some of these patients have been through multiple kind of experimental therapies, and they're at the end of the road. for their disease. And so if we were able to do this analysis earlier, could we perhaps help more patients? And, because these tumors, many of these tumors are already so heavily treated that, we know already that definitely these single agent therapies are not gonna help in a lot of the cases for these very heavily treated tumors. Although we were very surprised by that myoepithelial carcinoma case that responded to a single agent therapy.
So I found that I can't ask questions on the Q&A because I'm a panelist. So while we're waiting, is it okay if I ask a question?
Absolutely, please.
So for the, this is a kind of a technical question. For the patients who are coming in, not tumors, but you said you're starting to do patients in combination with exomes to see if you can find out what's
Yeah.
Are those patients, for the turnaround time for those, who again, turnaround time we think about a lot, do you have to grow up their cells and lymphocytes, or can you do RNA testing directly on the, do you have to cell culture lymphocytes, or can you do the RNA testing directly on them?
We can do, we've started a pilot project with UCSF. So UCSF has a exome program, so they do exome in their clinical lab, and then they have these patients that, exome negative, or exome has some VUSs that might be interesting, but you can't. And then, so for those patients, so we've been doing RNA sequencing on the blood sample the same sample that they use for exome. We know that, with gene expression, you have to worry about differences in expression in different tissues. So if a patient has a, CNS phenotype, would you see that in blood? Like, would you see that gene aberrantly expressed in the blood? Maybe not. So, but you cannot necessarily get access to the brain tissue. So we think about those things a lot. Our colleagues at UCSF suggested that maybe focusing on like neuromuscular disorders, for example, like somewhere where you can get access to the affected tissue would be useful, but then it's limited, it's neuromuscular disorders. I don't know what other group of disorders where you can biopsy the affected tissue, but kind of disorders like that would be especially.
And is it really dependent on having a good database of control tissue for the same tissue?
Absolutely, yeah. So that's key, having, something to compare to where you have data from multiple, ideally hundreds of people. And so with blood, we do have that. So we have in the GTEx data set, we have blood from, I think, 200 people right now, which is, more is always better, but at least we have that. But exactly to your point, so for affected tissue, we have to make sure that we have tissue samples to compare to that are from the same tissue.
Yeah, thank you.
Thank you for that question. That's great.
All right, we have another question. It's a bit long. Average expression data that one might get from extracting and evaluating RNA from a heterogeneous tumor sample could have diagnostic advantages and/or disadvantages. How do you think about this situation?
Yeah, so I think maybe the question is around tumor heterogeneity. So we are, definitely looking at bulk RNA sequencing data, which provides an average of what we see in the tumor, and we know that tumors are much more complex. There are immune there is different cell populations present. So we have started thinking about single cell sequencing data, and we have a pilot or pilot project where we have bulk sequencing data and also a single cell data from the same tumor. And we have a couple of data sets like that from glioblastoma, so brain tumors. And so we want to see, our gene expression outliers we're finding, are they specific to a particular cell population within that tumor? Or, ideally, if they're found in every tumor, that's not going to be the case. But yeah, so we want to look at that for sure to know, to address the heterogeneity. And of course, that will be different for different tumors. So we are starting with glioblastoma because we have those kind of data where we have bulk and single cell from the same tumor. And definitely, I think that's a limitation of this approach, that we are looking at the average signal from the tumor.
I don't see any other questions. So thank you so much, Dr. Vasky, and thank you, Dr. Eckel, for introducing us. This has been really wonderful to hear you, and hopefully you're open to other questions from people who may want to email you questions instead of asking.
Absolutely. I was just going to say, if others, if people want to email me questions, I can, there is no chat, but my email, well, I guess Dr. Eckel has my email. It's very simple. It's my first name, olena@ucsc.edu. So please ask me questions. Or if you would like to, if anybody has RNA sequencing data and would like to work together, I'd be open to that as well, especially if it's in pediatrics.
Great. Thank you.
Thank you very much for having me.